This is part 1 of Chapter 11's lecture on cell communication. So we're going to start just by looking at an overview of cell signaling and basically just what is its purpose, which comes down to communicating between cells to coordinate activities. Sometimes the communication is actually not just cell to cell, but it could be from outside of the organism, from a different organism's cells. For example, yeast use cell signaling for mating purposes. But all cell communication is going to use a signal transduction pathway in which a signal on the cell's surface is converted into a specific response within the cell, sometimes through a series of steps. The three parts of cell signaling are 1, reception, 2, transduction, and 3, response. So keep that in mind. That's an important breakdown of how cell signaling works. And Earl Sutherland is credited with coming up with this definition of cell signaling. You can see in this diagram down below here the reception, transduction, and response. So reception is often on the cell membrane. So from outside of the cell, a signal comes. And the transduction is simply how it gets through the cell to its destination where the response is going to occur. The messenger that comes and brings the information or the signal is a signal molecule. And it can come from locally or it can come from a long distance. In both types of signaling, there is a specific target cell that's going to recognize and respond to that specific signaling molecule. If it's come from close by, then it's called a local regulator, which means it's something that's going to influence the cells that are close by in the same vicinity. So things like growth factors or neurotransmitters are examples of local regulators. This local type of cell signaling is called paracrine or synaptic signaling. The other type, the long distance, is going to be using hormones and is called endocrine signaling. And so this is, of course, hormones, are made in one part of the body. They're secreted into the bloodstream and travel a long distance to get the response. Another type of communication is going to occur that's even closer than paracrine, and that's going to be direct contact. So we talked about this in class with one of our bell ringer questions about cell junctions. And cell junctions are actual connections and, I guess, tunnels, for lack of a better description, that connect two cells together. And so they can exchange all sorts of substances in their cytosol and pass them freely back and forth. So in an animal cell, this is called a gap junction. And in a plant cell, it's a plasmodesmata. Sometimes that communication that's close by is actually from just one surface molecule to another. So that's still direct contact that's occurring between the two cells, but it's by way of those proteins that are embedded in the cell membrane. We talked in class about the difference between a gap junction and a tight junction. And so a tight junction is not really a form of communication. It's really, for example, adjacent epithelial cells will be so close together at the apical surface of the cell that it actually has a network of proteins that are linking them together. And their purpose is to limit the passage of molecules and ions through the space between the cells, so that anything that needs to pass through is actually going to enter a cell by diffusion or active transport in order to pass through into the tissue. So this provides protection. It provides a tighter control over what kind of substances are actually going to be allowed through. Another type of connection between cells is called an adherens junction, and that's a strong mechanical attachment between two adjacent cells. And a good example of that is with cardiac muscle, which has to be held tightly together as the heart is expanding and contracting in a pretty violent way. And so these adherent junctions are actually this strong, narrow bands that are going to attach the two cells together and hold them together. And finally, the gap junctions are made up of proteins. They're 1.5 to 2 nanometers in diameter, and they're actual channels or tunnels that allow that free passage between cells. And their cylinders are actually constructed of 6 copies of a transmembrane protein that's called a connexin. In plant tissue, the desmosomes and the plasmodesmata are the ones that are allowing that free flow between the cell's cytosol, between the two different plant cells. Okay, so that signal transduction process begins, the first of the 3 steps is reception of a signal. So that reception is going to take place when the chemical signal binds to a cellular protein, which is usually going to be found on the cell surface or on the plasma membrane. But sometimes it's also found within the cell itself. So a signal molecule is going to be complementary in shape to a very specific site on the receptor, and it attaches there similar to the same action we see with enzymes. That lock and key idea that we have with enzymes where there's a specific molecule that fits a specific site. So whether the receptor is on the surface of the cell membrane or if it's inside of the cell cytosol, it's still going to have that same specificity. The molecule that does the attaching, so that signal molecule that's coming to bring the message, is called a ligand. It's a small molecule that's going to, again, bind specifically to the receptor site of another larger molecule. And again, most signal receptors are plasma membrane proteins that are going to transmit the information from outside the extracellular environment to the intracellular space inside the cell. Okay. So there are 3 major types of membrane receptors. We're going to look at all 3 of those. The first one we'll look at is a G protein linked receptor. And this is found on the plasma membrane. And there's a receptor site, like I've been describing, that's looking for a specific ligand. And so in this picture, it's the purple molecule that you see here. These types of receptors are very, very common in the human organism. They're involved with fetal development, they're heavily involved in vision, and also in the sense of smell. So, for example, there are about 1,000 different G protein linked receptors involved in our sense of smell alone. What they're made up of is one long protein that actually goes back and forth. It weaves back and forth through the plasma membrane 7 times. And as it's actually passing through the plasma membrane, it's in an alpha helix. So there are 7 alpha helices that make up a G protein receptor. Okay, let's get the next slide. And what's attached now to that receptor protein on the cytoplasmic side of the plasma membrane is the actual G protein. So this purple receptor protein is attached to this aqua-colored protein, which is called the G protein. When it's attached like this, it's in its inactive form. And when it's inactive, it has attached to it a GDP. But what happens when a ligand is attached to the receptor, that changes the conformational shape of this receptor protein, which then allows this protein, the G protein, to change shape. And in doing so, it actually goes through a phosphorylation and becomes GTP. And then at that point, that GTP makes it an active G protein. And what happens is now this G protein travels a short distance along the cytoplasmic side of the plasma membrane until it reaches a different target protein. In this case, it's going to be an enzyme, which when this G protein is bonded with the enzyme, causes the enzyme to become activated and do its job, which is the cellular response for this G protein. Now, this G protein actually is acting as a relay molecule because it's taking the signal from here and it's transferring it over to a target protein. Sometimes this target protein is going to be an enzyme, like I said before. It can also be an ion channel that becomes activated and opens up. The actual G protein is made up of subunits, and what's happening between this stage and this stage is once the GDP becomes GTP, it actually separates this G protein apart into its different subunits, and it's actually only one of the subunits that travels over and attaches to the target. The target of the dissociated component of that G protein is again going to either be an enzyme or an ion channel in the plasma membrane that's going to relay the signal onward to its final result. One ligand can activate many different receptor family members. For example, there are at least 9 distinct G protein linked receptors that can be activated by adrenaline. So adrenaline is a ligand or a signal molecule, and it activates these G protein linked receptors. There are another 5 different G protein linked receptors that can be activated by acetylcholine. And also serotonin acts on G protein linked receptors as well. It actually can act on 15 different G protein linked receptors. And it's worth noting that about half of all known drugs work through these G protein linked receptors. The next type of receptor that we find on the cell membrane is a tyrosine kinase receptor. So tyrosine, as you should know, is an amino acid, and so this receptor system is especially effective when the cell needs to trigger several signal transduction pathways and the cellular responses at one time. So for example, with cell growth, that's a time when you need a rapid response that gets amplified. And that's what it's talking about here in this step. It's specialized for triggering more than one signal transduction pathway at one time. We're going to look at the structure of these receptors in just a second here. First, you should understand what kinase does. So kinase is an enzyme that catalyzes the transfer of phosphate groups. And so a tyrosine kinase receptor belongs to a major class of plasma membrane receptors that have enzymatic activity associated with them. So you can tell with the ACE ending of the word here, kinase.